Welcome back to this next video, and in this video we are going to talk about the types of the gene therapies. In the first two parts of this video, we generally talked about the gene therapy, about the success cases of the gene therapy, and in this particular video we are going to talk about the different types of the gene therapy. One of the type of the gene therapy that is known as the somatic gene therapy. Now, as the name indicate, in somatic gene therapy, the therapeutic gene they are transferred into the somatic cells of the patient, that is the non-sex cells of the patient. In the somatic gene therapy, any modifications and effects that will be restricted to that individual patient only, because you are inserting the therapeutic gene into the somatic cells, and that will not be transferred into the later generation, because we all know that the characters they are transferred from one generation to another generation utilizing the sex cells. And in this particular somatic gene therapy, as you are focusing on the somatic cells, so any changes that will be restricted to that particular individual and will not be transferred to the later generation. This is just a general overview how the somatic gene therapy that works. For example, this is a patient. You take the cells from the patient, and these cells are having mutations of a particular gene, so you can call them as the mutant cells. You have got a vector having the gene of interest, that is that you have got the therapeutic gene in the vector. We'll be having a detailed discussion on the vectors in the next video. So what you do is that when you insert your gene of interest into the vector, you introduce the vector into these mutant cells. This vector is going to introduce the corrected copy of that gene of interest into these cells. You are going to grow these cells, and these altered cells, by altered I mean that these particular cells now have got the therapeutic gene. They are reinserted into the patient, and the condition of the patient that can be recovered. So this is known as the somatic cell gene therapy. If you talk about the somatic cell gene therapy, over 600 clinical trials they are underway in the United States, and many of these trials they are focusing on genetic disorders like immunodeficiencies, like the hemophilia and the cystic fibrosis. Now, these immunodeficiencies, the hemophilia and the cystic fibrosis, they are very good candidates for somatic cell therapy because they are caused by single gene defect. So, treating with the diseases that are caused by single gene defect as compared to the replacement of the multiple gene in somatic cells, it is very easy. Therefore, the gene therapy, currently the gene therapy is focusing on those particular diseases which are caused by the single gene defects. The other type of the gene therapy that is known as the germline gene therapy. Again, as the name indicate, in the germline gene therapy, what you are going to do is that you are going to insert the therapeutic gene into the germ cells, that is the sperm of the eggs. So you are actually modifying the sperms or the eggs, and when you are going to modify these germ cells, these therapeutic gene that will be integrated into their genomes. And as you all know that this sperm and egg, they are going to combine, they are going to make a zygote. That means that the zygote is going to give to make a complete human body. So the gene of interest that will be transferred to the next generation as well. So the germline gene therapy, it is going to modify the sperm and the egg cells, and these genetically modified cells, they are going to make a complete organism with the modified gene, and that will be heritable to the next generations. Now, the germline gene therapy, it looks very attractive, and in theory, it is highly effective in counteracting the genetic disorders and hereditary diseases. But the problem is that some of the major countries of the world, like the Australia, Canada, Germany, Switzerland, and the Netherlands, they prohibit this application in the human being, at least for the present, for technical and ethical reasons, including insufficient knowledge about the possible risks to the future generation, and as the germline gene therapy, the character that will be transferred to the next generation, so it is going to have a very high risk as compared to the somatic gene therapy. So currently in the world, most of the gene therapy, they are focusing on the somatic gene therapy as compared to the germline gene therapy, at least for the present, for technical and ethical reasons. This is another type of the classification of the gene therapy. The first type of the classification was based on the cell type that you are targeting, whether you are targeting the somatic cells or you are targeting the germline cells. This classification is actually focusing on the procedure that you are adopting for the gene therapy. The first type is known as the ex vivo gene therapy, and it is clear from the name that you are doing something out of the body. Therefore, we are using this term ex and vivo. You have got these copies of the therapeutic gene. You are going to insert those copies of the therapeutic gene into the viral vectors, and we will discuss that as I've told you. And then what you are going to do is that you are going to take the target cells from the patient, and you are going to grow the viral vectors in these target cell in the lab. So these viruses, they are going to insert the therapeutic gene into these cells that you have removed from the patient, and once the cells that have been modified with the therapeutic DNA, they are going to be reinserted into the patient body, and the therapeutic gene is going to produce a therapeutic DNA and hence therapeutic protein, thereby treating the disease condition. So this is known as the ex vivo. That means you are modifying the cells, you are inserting the therapeutic gene outside the body. Another type of the gene therapy that is known as the in vivo gene therapy, and as the name indicates, you are actually delivering the gene to inside of the body without removing the cells from the patient. So what you do is that you are going to take these your therapeutic gene, you are going to insert that into the vector, and you are directly going to insert the vector into the human body without removing the cells from the patient body. So this is known as the in vivo gene therapy. We will continue this discussion in the next video.